Okay, thank you so much. Then let me share the screen. Then first of all, I would like to thank our colleagues, particularly Korean colleagues, to giving this opportunity for me to speak. And today I would like to talk on the global blue ocean metagenomics, but actually I like to talk on the whole metagenomic project. So in fact, I would like to propose the international metagenome research collaboration. Actually, this is Japan Island, and I worked for marine metagenomics almost for 15 years, then including the sampling and sequencing, but mostly we work on annotation, particularly functional annotation, and then like microbial diversity, how it changes over the time, particularly along the Japan Island. Then also we constructed the marine metagenome database when I was in Japan. Then since I moved to Saudi Arabia, already I have been 8 years so far. Then it was a great opportunity to make a comparative metagenomics between Red Sea along Saudi Arabia, particularly I think west side, then the metagenomic data in Japan. But now, since we accumulated the so huge metagenomic data, particularly ocean data, we decided to make a gene catalog, including all possible ocean data, marine data. So therefore, the theme is very simple. I think we like to construct the gene catalog in a marine domain, how many genes there, also so-called MAG, metagenomic annotated the genome, maybe MAG can be constructed. So just like you have pieces of a logo, then we like to make a classification. Those are like a gene catalog, essentially, depending on the functional structure, functional categorization. This is simply the advancement, research advancement history of metagenomics, like particularly ocean microbes started from 2004, then maybe human microbes later, but now really a huge accumulation of metagenomic data are available, including publicly available data. So first we are focusing on the marine or ocean metagenomics, but later on we include all global metagenomics. Then purpose is very, very simple. Just we like to make a gene catalog. So therefore, we call brute force or brute computation. We utilize our supercomputer. Then, actually, those metagenomic data, whatever it is, actually first we worked on marine metagenomics. Then later we constructed the much huge data. So then the pipeline we call a KAUST metagenomic annotation platform, KMAP. I think we constructed input annotation process, visualization on the comparative analysis. Those are not really unique, but maybe more consistent or coherent to make a functional annotation. Particularly when you have, this is our map, DMAP. Now it is, first we call the DMAP, then we changed KMAP. Then what kind of functional categorization, how those are mapped? Actually red are not really mapped. So green are mapped. So from this point of view, our gene catalog are quite rich. And we already published like a KAUST metagenomic analysis platform, okay, rather than annotation analysis platform. Already we published a paper, and then kind of pie chart or like even phylogenetic tree, then gene network. Those annotations are made actually, but I don't have enough time to explain all of them. And when you pay your attention to ocean genome, those are kind of large ocean expedition. Then they took samples here. Those are kind of sampling distribution. For example, like Malaspina Deep Ocean project is the blue, and like a Tara Ocean project has been designated the pink, which made a tremendously huge number of sampling from shallow water to deep water. So, and also we published deep ocean microbes here. That's very interesting features. And also from deep sea gene catalog, at that time still not full scale of our gene catalog depository, but we found that plastic degrading enzyme. So therefore, those are very important contribution, particularly we call bioprospecting, how we can dig out the variable genes from those huge data. So huge data is like, crude oil field or like a mining, and actually, literally, I think the data is becoming such asset. Then in a KAUST, we conducted the mesocosmos, which means we create artificial pond in the sea. Then we can put nutrient. Then how microbial community respond. This is experimental metagenomics. Then we can extract kind of elementary process or elementary factor to understand how actual wild microbial communities are moving. Okay, then this is like we published those Metacosmos, and here, sorry, this is a different topics, but we utilized K-map pipeline for annotating SARS-CoV-2 genes or genomes, and we have published one paper for COVID-19 mutation tracker in a Lancet pathogenetic disease. Then now we can have a real-time, almost real-time variant tracing system every day. For example, this is as question was made, this is Omicron. Just 2 days ago's genome data. Now we have more than 530 Omicron genomes. And those are Delta, and totally quite different. And according to annotation through our K-map, I think that those new Omicrons seems to be accumulating somewhere, particularly certain individual who is known to be a survivor or retainer of viruses, which means usually COVID-19 viruses will be eliminated from the one individual unless it is infected other people. Someone can retain the viruses for a long time, even 2 months or 3 months. Then the human body is making a role of culture media for these viruses, then accumulating a huge number of mutations. Then once it infected other individual, then again a mutation is taking place. So it's a totally different, but certainly there is another hypothesis. This may have come from animals, not human, but certainly Dr. Yushi Lee explained it would be very important to understand the origin of emergence. Sorry, a little bit different story, but let us go back to original story. Then I think our analysis was expanded from marine metagenomics to all metagenomics, biosphere metagenomics. Particularly by utilizing our pipeline gene catalog, we can search for new antibiotics from those gene catalogs. Of course, we are interested in the evolutionary implication of those huge number of genes, but so therefore, this is the same field, but we have metagenomic samples, assembles, and the full-length gene catalogs and the extensive annotation, then data sharing. Of course, I think there are a lot of questions, a lot of issues, how we can define a gene or how we can categorize. But maybe just aside from such a heated discussion, anyway, why we don't have a large picture of our gene world on the globe. So this kind of a gene catalog already, I don't have enough time to explain a lot, but those are kind of a sampling point as an x-axis, then y-axis shows how many genes are there. So far, we have 275 million genes. That's a huge asset as a gene catalog. So as I said, it becomes actually genetic resources already. When you look at the samples, we collected more than 30,000 metagenomics. Then this is the newest one. Then we have now 3 billion gene catalog. And if we can define representative gene, which means excluding redundancy, still we have 1 billion representative genes. And in terms of annotated genes, 0.6 billion gene catalogs. Such a huge gene pool can be elucidated, classifying those genes. This is a million, therefore, host-associated genes, 244 million genes, like a land and the ocean, almost 204 million genes, land 137 genes. So biome, this is a gene catalog, but if we have make metagenomic annotated genome, then we can see the number of gene cluster, not only single gene cluster, particularly antibiotics is produced from a metabolic pathway, not single gene. Therefore, we have to have metabolic pathways to produce particular metabolite, which can be utilized antibiotics. Before that, even we can search for cellulase, which is very important for bioenergy, lipase, which is also important for detergent. So those are industrially very useful enzymes can be found such a huge gene catalog data set. So this is rather like a MAC, metagenomic assembled genomes. So therefore, more not gene catalog, more like a genome itself. Certainly, we can see how those are distributed. According to our statistical analysis, still number of gene catalog or genes or mega not saturated. So therefore, once as such a expedition project has advanced, we should be able to get more number of genes. So how many genes do we have on the Earth? That's a very interesting question. And here, like examples, like a biosynthetic gene cluster, which shows antibiotic like NRPS, PKS. So once we have like a metagenomic assembled genomes, as I said, MAG, then we can see such a gene catalog or gene cluster structure. From here, we can infer not only gene enzyme itself or protein itself, we can have kind of a system such as new genome editing CRISPR-Cas9 system, which is totally different from existing system. So this is just like we mentioned the big data in the life science, and we published the marine metagenomic data as a vision. As I said that we have constructed kind of a V. It has been so large on the ocean, but now it becomes much, much smaller. But what we are saying is like sensors or ship, then satellite can be utilized to analyze those data. In fact, we have ALE. This is a satellite venture company in Japan. They now working on satellite, and I had misunderstood that launching a satellite is so expensive, like $100 million or even $500. But the reality, it's not. It's very cheap. Like I don't say cheap, but like even $200,000, you can have your own sensor or satellite. So I think time has come. The satellite is like my satellite, like not the national center. Of course, NASA, JAXA, whatever. It's a much more R&D, but actual industrial satellites is moving. So since I'm in a director of Marine Ocean Institute in Japan, so therefore collaborating with these venture companies, we are now trying to surveillance on the sea around Japan, and there are two ways. One is geostationary satellite, which is like a 20 to 25,000 meters high, but this is a stationary orbit. Therefore, once a day you can observe. On the other hand, smaller satellite, which is much cheaper, which can be on the orbit, like 5,000 or 6,000 meters away. Therefore, this is much closer, but because of not geostationary, it's not really stable. On the other hand, those smaller satellites can compose so-called constellations, like 10 or 15. Those number of smaller satellites can work all together in an organization. So therefore, we are now planning to utilize constellation-forming satellite surveillance on the particular sea. And so they also collect atmospheric data, not only the air, but close to the ocean. And since we have a technology of so-called digital DNA chip, which is our memory in the computer, we can have probes. Then any data of metagenomics can be hypothetically or computationally hybridized. Then from this pattern, recognized by AI, we can identify what it is. So the proposal is we can create more marine environmental blockchain, and actually, like we can have a given place, particularly ocean, like depending on the depth, we can have DNA chip. Hypothetical DNA chip, data chip can be accumulated. So in collaboration worldwide, we should be able to map of those regions. Even we can make a surveillance of a nice fishery field too. And actually, United Nations now launched 1 decade, 10 years for ocean science from last year. It will be a very good, important project. So conclusion, marine metagenomics is powerful, but we not only ocean, we constructed the whole biosphere metagenome, particularly focusing on gene catalog. And the digital DNA chip can be utilized for pattern recognition by AI, and the so-called, we call marine environment blockchain network is proposed as a vision, and the international collaboration is essential. So those are our team, and actually our center, when I got the director, only 50 to 60 people. Now I'm so pleased to announce that we are grown about 200 people. 200 people are working in our center. So, okay, thank you so much. Thank you so much, and particularly Dr. John Berg. Thank you for allowing me to speak before you. Thank you.
Okay, thank you.